ನಿಮ್ದು ಇಂಟ್ರೋ ಮುಗ್ದಿ ಫೇಸ್ ಬಂದ್ಮೇಲೆ
ಅವಾಗ ಹೋಗ್ಬಿಟ್ಟಿತ್ತು, ಬಟ್ ಇದು ಓದ್ತಿದ್ದೆ ಬಂತು.
ಏನು ಓದೋದು ಬೇಡ, ಹೋಮಿಗೆ ಹೋಗಿ ವೈಟ್ ಬೋರ್ಡ್ ಅಂದ ತಕ್ಷಣ ಬಂದ್ಬಿಡುತ್ತೆ. ಆಯ್ತಾ? ನಿಮ್ಮ ಮುಖ ಬಂದ ಕ್ಯಾಮ್ರಾ ನೋಡ್ತೇವೆ. ಬೇಗ ಬರ್ತದೆ.
Press the bell icon on the YouTube app and never miss another update.
Namaste children, myself Dr. Usha Diya. Today I am going to take introductory class on the chapter The Cell, Basic Unit of Life. The cell, the fundamental unit of life. Okay. So we'll go with the chapter. Cell, the unit of life. Okay. See, when we look around us, and when we look the surroundings, we see both living and non-living. Eradu nodteve, living and non-living. So then the question arises. So what is the difference between the living and non-living? Living irodu, non-living yaud illa. Understood? So what makes the organisms to live on this earth? So havaga nodaga, the answer is, it is nothing but cell. It's nothing but cell, which is present only in living organisms and it is not present in non-living organisms. Inanimate andre non-living things. So these cells are not present in inanimate things or non-living things. It is present only in living organisms. Presence of this makes the living organisms to survive on this earth. Understood? So cell is the basic unit of life. Cell is the basic unit of life. Cell is considered as basic unit of life. After knowing the cell, it is responsible for life on this earth, existence of life on this earth, we grouped organisms into two categories. Organisms, living organisms na group ಮಾಡ್ತಾ ಇದೀವಿ into two categories. ಯಾವ್ಯಾವುದು? So based on number of cells, organisms are categorized into unicellular and multicellular. Unicellular organisms and multicellular organisms. Understood? So we grouped organisms into two categories. One, unicellular. Unicellular means what? They are made up of single cell. Multicellular means they are made up of many cells. Understood? The organisms in which the single cell is responsible for its existence and performing all the essential functions of life, we consider it as unicellular. Whereas in multicellular group of cells, they form tissues, organs and organ system for the survival of that organism. Those come under a group called multicellular organisms. Understood? So unicellular, we know that example for unicellular is bacteria, amoeba, etc. They all come under unicellular. Whereas in multicellular, human beings, we are all come under multicellular organisms. Understood? So after knowing this, unicellular and multicellular. We'll go with now who discovered the cell for the first time. Understood? So it was Robert Hooke. Robert Hooke in the year 1665. Oren Matare, it took cork cells from the plant. It took cork cells which are dead cells. Cork cells are dead cells. Hagagi, when he was studying these cells, that time microscopes were not very well developed. Electron microscopes avayavu irlilla. So by using simple microscope, he studied these cork cells and he observed only this a wall. Understood? Small size compartment which is surrounded by a wall. Inside that there was no any other structures. ಹಾಗಾಗಿ ಅವರು ಅದಕ್ಕೆ ಏನಂತ ಹೆಸರು ಕೊಡ್ತಾರೆ, ಸೆಲ್ ಅಂತ. ಎಂಟಿ ಕಂಪಾರ್ಟ್ಮೆಂಟ್. ಎಂಟಿ ಕಂಪಾರ್ಟ್ಮೆಂಟ್ he called it as a cell. He called it as a cell. ಅರ್ಥ ಆಯ್ತಾ? ಅವ್ರೇನಾದ್ರು ಲೈವ್ ಸೆಲ್ ತಗೊಂಡಿದ್ರೆ maybe he would have given us the cell structure and it is made up of different types of organelles atara. But unfortunately ಅವ್ರು ತಗೊಂಡುವಾಗ cork cells ತಗೊಂಡ್ರು, they are dead cells. ಹಾಗಾಗಿ ಅವ್ರು when he observed these cells under microscope, ಅವ್ರಿಗೆ ಏನು ಕಾಣ್ತು, it is a small compartment surrounded by a wall. For that he called it as a cell ಅಂತ ಹೇಳ್ತಾರೆ. Understood? After this, the other scientist, Anton von Leeuwenhoek, he described first live cell. ನಮಗೆ ರಾಬರ್ಟ್ ಹುಕ್ ಕೊಟ್ಟಿದ್ದು ಯಾವುದು, ಡೆಡ್ ಸೆಲ್ ಕೊಡ್ತಾರೆ. he told only the cell is a compartment, inside that empty space is present. But Anton von Leeuwenhoek gave us live cell. ಯಾಕೆಂದ್ರೆ by the time he studied the cell, they very well developed electron microscope were there. Due to that advanced in microscope, he was able to study the live cells in an organism. Live cells in an organism. So first person who gave us live cell is Anton von Leeuwenhoek. Understood? So like this, many scientists went on studying about the cells. So one day the Matthias Sheldon, in the year 1838, he was a botanist. And one more person, Theodore Span, in the year 1839, he was a zoologist. So ಇವ್ರು ಇಬ್ರು ಏನ್ ಮಾಡ್ತಾರೆ, see, Matthias Sheldon, botanist ಅಂದ್ರೆ the person who studied only about plants. Understood? Zoologist ಅಂದ್ರೆ the person who studied only about animals. So Matthias Sheldon ಬರೀ plants ಗಳನ್ನ ಸ್ಟಡಿ ಮಾಡಿ ಏನ್ ಹೇಳ್ತಾರೆ, all plants are made up of different types of cells that make up tissues and organs ಅಂತ ಹೇಳ್ತಾರೆ. Matthias Sheldon ಯಾವ್ದು ಸ್ಟಡಿ ಮಾಡ್ತಾರೆ, he studied only the plants. After studying plants, he told all plants are made up of different types of cells. All plants are made up of different types of cells which form a tissues and organs and organ systems ಅಂತ. So plant tissues we studied 9th ಅಲ್ಲಿ ನಾವು ಓದಿದೀವಿ meristematic tissues and permanent tissues. Meristematic tissues and permanent tissues. Permanent tissues ಅಲ್ಲಿ we studied parenchyma, collenchyma and sclerenchyma. Understood? So, ಇವರೇನು ಹೇಳ್ತಾರೆ, all plants are made up of different types of cells which form tissues like permanent and meristematic tissues. Under permanent we have parenchyma, collenchyma and sclerenchyma, and complex permanent tissues like xylem and phloem. All these cells and tissues form a plants ಅಂತ ಹೇಳ್ತಾರೆ ಯಾರು, ಮ್ಯಾಥಿಯಸ್ ಶೆಲ್ಡನ್. ಅದೇ ರೀತಿ Theodore Swan studied animal cells. When he studied animal cell, he taught all animal cells have an outer limiting membrane. All animal cells have outer limiting membrane. Now we are calling it as, ಅದನ್ನ ನಾವು ಇವಾಗ ಏನಂತ ಕರೀತಿದ್ದೀವಿ, plasma membrane ಅಂತ ಹೇಳ್ತೀವಿ. We are calling it as plasma membrane. Again he studied plant cells also. ಅವ್ರು ಸುಮ್ಮನೆ ಇರಲ್ಲ. Again he studied plant cells and he told even the plant cells have a unique structure called cell wall. Inside to cell wall we see a cell membrane. Cell wall is unique to plants ಅಂತ ಹೇಳ್ತಾರೆ ಯಾರು, Theodore Swan. Understood? After studying about both the plant cells and animal cells, and Theodore Swan gave one hypothesis. ಇವರು ಎರಡು ಅನಿಮಲ್ ಸೆಲ್ ಪ್ಲಾಂಟ್ ಸೆಲ್ ಎರಡನ್ನೂ ಸ್ಟಡಿ ಮಾಡಿ ಒಂದು ಹೈಪೋಥಿಸಿಸ್ ಕೊಡ್ತಾರೆ. ಏನಂತ, all plants and animals are made up of cells and products of cells ಅಂತ ಹೇಳ್ತಾರೆ ಯಾರು, Theodore Swan. He gave us one hypothesis. Hypothesis ನಮಗೆ ಏನು ಹೇಳ್ತು, all plants and animals are made up of what, cells and products of cells ಅಂತ ಹೇಳ್ತಾರೆ. So after that both studies are combined and they gave us a theory called cell theory. ನಮಗೊಂದು theory ಕೊಡ್ತಾರೆ ಇಬ್ರು ಸೇರಿ, it is cell theory. So first point, cell theory includes what, all living organisms are made up of cells and products of cells. Cells and products of cells. But they fail to give us how the new cells are going to be formed. Or ಏನು ಹೇಳ್ತಾರೆ, whether it is plants or animals, they are made up of group of cells and products of cells ಅಂತ ಹೇಳಿದ್ರು. ಆದ್ರೆ ನ್ಯೂ ಸೆಲ್ ಯಾವ ರೀತಿ ಫಾರ್ಮ್ ಆಯ್ತು ಅಂತ ಹೇಳಿಲ್ಲ. Understood? That was given us by Rudolf Virchow. Rudolf Virchow in the year 1855. ಇವರು ನಮಗೆ ಒಂದು he gave us one statement that hominis cellula e cellula. Hominis cellula e cellula ಅಂತಾರೆ. ಸೊ ಅದು ನಮ್ಗೆ ಏನು ಹೇಳ್ತಿದೆ ಅಂದ್ರೆ, all new cells are going to be formed from pre-existing cells ಅಂತ ಹೇಳಿ. All new cells are going to be formed from pre-existing cells ಅಂತ ಹೇಳಿ. Who told that? Rudolf Virchow. ಯಾವುದೇ ಒಂದು ಹೊಸ ಸೆಲ್ಲು it is going to be formed from pre-existing cells ಅಂತ ಹೇಳಿದ್ಮೇಲೆ cell theory modify ಮಾಡಿಬಿಟ್ಟು we included that point also here, that new cells will form from pre-existing cells. So now the cell theory is complete. So Matthias Sheldon and Theodore Swan, they told us all living organisms are made up of cells and products of cells anta helidu. But they fail to give us how the new cell is going to be formed. Adunna complete madid yaru? Rudolf Virchow. He gave us a statement that how many cellula is cellular. That means new cells are going to be formed from pre-existing cells anta helidu. So adunu ನಾವು include ಮಾಡಿಕೊಂಡು cell theory na update ಮಾಡ್ಕೊಂಡ್ವಿ by telling that all living organisms are made up of cells and products of cells, and new cells will form from pre-existing cells. Understood? So after knowing about the cells, we'll go with the difference between prokaryotes and eukaryotes. Okay. So prokaryotes, nothing but they are the cells which have outer layer, that is cell wall. All prokaryotes have an outer layer called cell wall. Some bacteria will have another layer around the cell wall. It is nothing but capsule. It forms a structure called capsule. But it is not seen in every prokaryotes, but it is seen in bacteria. But in all prokaryotes we see cell wall. Inside to the cell wall we see cell membrane. Inside to this cell wall we see a structure called cell membrane. So this is cell membrane. Understood? Attached to this cell membrane we see a structure called flagella. If flagella is present, it helps in motility of the prokaryotic cells. Motility of prokaryotic cells. Some other appendages are there. They are attached to the cell wall. Smaller are called fimbriae. A little bit larger are called pili. But this will mainly help in adhering. Adhering ಅಂದ್ರೆ ಯಾವುದಾದ್ರೂ ಒಂದು ಸಬ್ಸ್ಟ್ರಾಟಮ್ ಮೇಲೆ ಹೋಗಿ ಅಟ್ಯಾಚ್ ಆಗಕ್ಕೆ ಹೆಲ್ಪ್ ಮಾಡುತ್ತೆ. ಯಾವುದು? ಫಿಂಬ್ರಿಯಾ ಅಂಡ್ ಪಿಲ್ಲೈ. But this is called flagella. Flagella helps in locomotion. So see here, in prokaryotes the DNA is naked. It is not surrounded by any nuclear membrane. Understood? And we see other structure called ribosomes, and one more structure called inclusion bodies are there. In some bacteria we see a circular structure called plasmid. Understood? Then we'll go with the eukaryotic cells. cell membrane. This is also cell wall. This is cell wall. Nuclear membrane. And we see ribosomes here also. See, now we'll study the difference between prokaryotes and eukaryotes. Okay, in prokaryotes, the outer membrane is cell wall. Understood? But capsule, it may be present in some bacterial cells. In most of the prokaryotes, the outer membrane will be the cell wall. Inside to that cell wall, we see a membrane called cell membrane. So cell wall is common to prokaryotes and plant cell. So this is plant cell. And this one is animal cell. Okay. Cell wall is seen in plant cell and in all prokaryotes, whereas cell wall is not seen in animal cell. Adadmele, inside the cell membrane is common to both prokaryotes and eukaryotes. Similar in structure and function also same in prokaryotes and eukaryotes. So the next class in Udana, what's the function of cell membrane? Cell membrane structure in prokaryotes and eukaryotes remains the same. Even the function remains the same. Inside to that prokaryote, we see a semi-fluid structure called cytoplasm. Cytoplasm. We see a fluid-like structure called cytoplasm. Here also the cytoplasm is present. In the cytoplasm, metabolic reactions occurs. What happens in cytoplasm? Metabolic reaction occurs which is responsible for the existence of life. What is metabolism? Chemical reactions that occurs in cell. Andre digestion, that is breaking of larger molecules into simpler form, or forming of simpler larger molecules by using simpler form, that is anabolism and catabolism. So that we study in your next chapters. Metabolism and the Egan class, it is a chemical reaction which occurs in cytoplasm of the cells, both in prokaryotes and in eukaryotes, which is mainly responsible for existence of life and the okay. Inside the cytoplasm, cellular organelles are present. Understood? But in prokaryotes, cellular organelles are absent, whereas in eukaryotes, cellular organelles like mitochondria, Golgi apparatus, chloroplast, ribosomes, all these cellular organelles are present inside the cells in eukaryotes, whereas cellular organelles are absent in prokaryotes. Only the organelle, non-membrane bound organelle, yaudadu ribosomes, are present in both prokaryotes and eukaryotes. Nodi, prokaryotes and mitochondria, Golgi apparatus illa bardilla because they are not seen in prokaryotes. They are seen only in eukaryotes. ಎರಡರಲ್ಲೂ ನಾವು ನೋಡುವಂತ ಆರ್ಗನಲೆ ನಾನ್ ಮೆಂಬ್ರೇನ್ ಬೌಂಡ್ ಆರ್ಗನಲೈಸ್ ಅಂತ ಅಂದ್ರೆ ribosomes. Ribosomes are seen in both prokaryotes and eukaryotes. Prokaryotes alli the ribosome size is 70S, whereas in eukaryotes it is 80S. Ashtu difference between ribosomes are seen in both prokaryotes and eukaryotes. Other difference between prokaryotes and eukaryotes andre, in prokaryotes, DNA is naked. Why it is naked? Because it is not surrounded by any nuclear membrane. You'll note it. DNA is surrounded by nuclear membrane. Understood? Nuclear membrane is absent in prokaryotes, but it is present in eukaryotes. That's why in prokaryotes, DNA is called naked DNA. Why? Because nuclear membrane is absent. Understood? The next, we see inclusion bodies in prokaryotes, which acts as a reserve food material. Other important structure what we see in prokaryotes is plasmid. This is a plasmid. Extra chromosomal DNA. This is a main DNA. This is very much required for prokaryotes and which transfer from parents to offsprings by carrying parental characters to their offspring. ಅದರ ಜೊತೆಗೆ ಇನ್ನೊಂದು DNA ಇದೆ, circular structure called plasmid. That plasmid we not seen in eukaryotes. It is present in prokaryotes that we used in biotechnology. You second year chapter ಓದ್ತೀರಾ. What's the use of these plasmids in biotechnology? Understood? So now when we see the difference between prokaryotes and eukaryotes, we can tell one more point, means prokaryotes are smaller in size, hagagi division is rapid in prokaryotes. Eukaryotes, they take some time for cell division, but here the rapid cell division we see in prokaryotes. Understood? That's the difference between prokaryotes and eukaryotes. ಇಲ್ಲಿ ನಿಮಗೆ write the difference between prokaryotes and eukaryotes ಅಂತ ಅಂದಾಗ first point ಏನು ಬರೀತೀರಾ? Nuclear membrane is absent and DNA is naked. ಇಲ್ಲಿ ಏನು ಬರೀತೀರಾ? Nuclear membrane is present which is surrounded by the nuclear membrane is present. Inside that we see a presence of DNA. The next point, cellular organelles inside the cytoplasm is absent except for the ribosomes. Illen baritira cellular organelles like mitochondria, chloroplast, Golgi apparatus, vacuoles, they are present. Understood? Organelles are present here, but organelles are absent. The next you can write plasmids are present in prokaryotes. Here the plasmids are absent. So like this you should write the difference between prokaryotes and eukaryotes. ಇಲ್ಲಿ ನಿಮಗೆ ಬರೀ prokaryote ಅಂತ ಬಂದಾಗ flagella helps in motility. Motility ಅಂದ್ರೆ locomotion. Whereas fimbriae and pillae helps in adhering of that cell into the substratum. ಎಲ್ಲಿ ಹೋಗಿ ಅಂಟ್ಕೊಳ್ಳುತ್ತದೆ, ಎಲ್ಲಿ ಹೋಗಿ ಇರುತ್ತೋ ಆ ಸರ್ಫೇಸ್ ಗೆ ಅಂಟ್ಕೊಳ್ಳೋಕೆ ಹೆಲ್ಪ್ ಮಾಡುತ್ತೆ, ಅಡೇರ್ ಮಾಡೋಕೆ ಹೆಲ್ಪ್ ಮಾಡೋ ಸ್ಟ್ರಕ್ಚರ್ಸ್ ಯಾವು? fimbriae and pillae. Whereas flagella helps in locomotion. And one more modification we see here is this plasma membrane, infolding of plasma membrane we see in prokaryotes, which helps in respiration and cell division. Mesosomes, these are called as mesosomes, which mainly helps in respiration and cell division. Understood? Coming to this inclusion bodies, this reserve food, that is a reserve food material. Ali glycogen store ಮಾಡ್ಕೊಂಡಿರುತ್ತೆ, phosphate store ಮಾಡ್ಕೊಂಡಿರುತ್ತೆ, which when the prokaryotes required energy, they utilize this stored food in the form of inclusions. Understood? That's the difference between prokaryotes and eukaryotes. Understood? So now we'll go with revision of this class. We came to know that all plants and animals which are living are made up of cell. The basic unit of life itself is a cell. Hagagi cell we are considering as structural and functional unit of life. Which one is structural and functional unit of life? Cell is considered as structural and functional unit of life. After knowing the cell, organisms are grouped into two categories. If they are made up of single cell, they are called as unicellular. If they are made up of many cells, they are called as multicellular. Cell anta antashna adralli ಏನೆಲ್ಲ ಕೆಪಾಸಿಟಿ ಇರಬೇಕು, independent existence ಇರಬೇಕು, and independently it should perform all the essential functions for the survival. ಅದನ್ನ ನಾವು ಸೆಲ್ ಅಂತ ಹೇಳ್ತೇವೆ. Any structure lack of this will not exist as a cell. It will not exist as a independent. Understood? ಅದಾದ್ಮೇಲೆ ನಾವೇನು ರಿವೈಸ್ ಮಾಡಿದ್ವಿ? Who gave us cell for the first time? Robert Hooke. In the year 1665 ಅಲ್ಲಿ it took cork cambium plant ಒಂದು ಸ್ಯಾಂಪಲ್ ನ ತಗೋತಾರೆ, ಅದು ಡೆಡ್ ಸೆಲ್ ಇರುತ್ತೆ. ಹಾಗಾಗಿ ವೆನ್ ವಿ ಅಬ್ಸರ್ವ್ ದಟ್ ಸೆಲ್ಸ್ ಅಂಡರ್ ಸಿಂಪಲ್ ಮೈಕ್ರೋಸ್ಕೋಪ್, ಅವ್ರಿಗೆ ಏನ್ ಕಾಣ್ತು, ಜಸ್ಟ್ ಎಂಟಿ ಕಂಪಾರ್ಟ್ಮೆಂಟ್ ಕಾಣಿಸ್ತು. ಹಾಗಾಗಿ ಅವ್ರು ನಮಗೆ ಅದನ್ನ ಸೆಲ್ ಅಂತ ಕನ್ಸಿಡರ್ ಮಾಡಿ ಸೆಲ್ ಅಂತ ಹೆಸರು ಕೊಡ್ತಾರೆ. Then the other scientists like Theodore Swan, who was a zoologist, ಹಾಗೇನೆ ಮ್ಯಾಥಿಯಸ್ ಶೆಲ್ಡನ್, ಬಾಟನಿಸ್ಟ್, ಇವರು ಇಂಡಿವಿಜುವಲ್ ಆಗಿ ಸ್ಟಡಿ ಮಾಡಿ ಇಬ್ರು ಸೇರಿ ಒಂದು ಸೆಲ್ ಥಿಯರಿ ಕೊಡ್ತಾರೆ. ಏನಂತ, ಆಲ್ ಲಿವಿಂಗ್ ಆರ್ಗಾನಿಸಮ್ಸ್ ಆರ್ ಮೇಡ್ ಅಪ್ ಆಫ್ ಸೆಲ್ಸ್ ಅಂಡ್ ಪ್ರಾಡಕ್ಟ್ಸ್ ಆಫ್ ಸೆಲ್ಸ್ ಅಂತ ಹೇಳ್ತಾರೆ. ಬಟ್ ದೇ ಫೇಲ್ ಟು ಗಿವ್ ಅಸ್ ಹೌ ದ ನ್ಯೂ ಸೆಲ್ಸ್ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಬಿ ಫಾರ್ಮ್. ಯಾವ ರೀತಿ ಹೊಸ ಸೆಲ್ ಫಾರ್ಮ್ ಆಗುತ್ತೆ ಅಂತ ಹೇಳಿಲ್ಲ. ಅದನ್ನ ನಮಗೆ ಯಾರು ಹೇಳಿದ್ದು, ರುಡಾಲ್ಫ್ ವರ್ಚ್ಯೂ ಹೇಳ್ತಾರೆ. ಏನಂತ, ಎವ್ರಿ ನ್ಯೂ ಸೆಲ್ಸ್ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಬಿ ಫಾರ್ಮ್ ಬೈ ಪ್ರೀ ಎಕ್ಸಿಸ್ಟಿಂಗ್ ಸೆಲ್. ಅದರಿಂದ ಏನ್ ಪೇರೆಂಟ್ ಸೆಲ್ ಇಂದ ಡಾಟರ್ ಸೆಲ್ಸ್ ಫಾರ್ಮ್ ಆಗುತ್ತೆ, ದಟ್ ಇಸ್ ಪ್ರೀ ಎಕ್ಸಿಸ್ಟಿಂಗ್ ಸೆಲ್ ಅಂತ ಹೇಳಿ ಆಮಿನಿ ಸೆಲ್ಯುಲಾ, ಈ ಸೆಲ್ಯುಲಾ ಅಂತ ಹೇಳಿ ಕೊಡ್ತಾರೆ ರುಡಾಲ್ಫ್ ವರ್ಚ್ಯೂ. ಸೊ ಸೆಲ್ ಥಿಯರಿ includes both the statements given by Rudolf Virchow and also Matthias Sheldon and Theodore Swan. After knowing the cell theory, so now we discuss the difference between prokaryotic cells and eukaryotic cells. So prokaryotic cells are smaller in size and they show rapid cell division. Prokaryotic cells outer layer is cell wall. Inside the cell wall we see cell membrane, which is common to both prokaryotes and eukaryotes. Cell wall is seen in plant cells of eukaryotes. Inside to the cell membrane in both we see a cytoplasm, fluid-like structure called cytoplasm, where the metabolism occurs, which is very much important for the survival of organisms. So metabolism anta andre alli formation of molecules ಆಗ್ತಾ ಇರಬಹುದು ಅಥವಾ ಬ್ರೇಕ್ ಡೌನ್ ಆಫ್ molecules ಆಗ್ತಾ ಇರಬಹುದು, which gives us energy. Understood? And cellular organelles are absent in prokaryotes like mitochondria, Golgi apparatus, chloroplasts, like that. Cellular organelles are present inside the eukaryotes. And one more structure which is common to both prokaryotes, eukaryotes anta nodaga, it is a ribosome. Ribosome is seen in both prokaryotes and eukaryotes. Understood? The next, the DNA is not surrounded by nuclear membrane in prokaryotes. ಅದಕ್ಕೆ ಇದನ್ನ ಇಲ್ಲಿ ನಾವು naked DNA is present in prokaryotes ಅಂತೀವಿ. Eukaryotes ಅಲ್ಲಿ DNA is surrounded by nuclear membrane. Understood? And inclusion bodies are present, which acts as a preserved food materials. They are present in prokaryotes. And one more structure modification we see in plasma membrane is mesosome, which mainly helps in respiration and cell division. That type of structures are not seen in eukaryotes. Understood? That's the difference between prokaryotes and eukaryotes. So you can, after knowing about the cell, we should know about which is the smallest cell and which is the largest cell. The smallest cell we consider as mycoplasma. Mycoplasmas are smallest cells, which is having 0.3 micron meter in length. The largest cell is mycoplasma cells are smallest cell. Largest cell is egg of ostrich. Ostrich bird nodidiralla. So egg of ostrich bird is the largest cell. Understood? So this is about today's class. In next class we'll discuss the difference between plant cells and animal cells and the function of every organelle present inside the cells. Okay. Thank you.